Hello everyone, welcome back to the session. So in today's class we are going to discuss about same thing, cellular respiration, just like yesterday, but in cellular respiration we have discussed about 4 stages and we have completed 1 stages in the previous class itself, that is nothing but a glycolysis. And in today's class we are going to discuss about this Krebs cycle. So this will be the second stage. This is not a second stage, but before that, see here, what are all the 4 stages we have discussed under the cellular respiration? First cycle will be the glycolysis, and the second stage will be the pyruvate decarboxylation, and this is called as a link reaction, which is the one linking the glycolysis and the Krebs cycle, which is the third stage of the cellular respiration. And the fourth and last stage of the cellular respiration will be this electron transport chain. So in today's class we are going to discuss about this pyruvate decarboxylation and the Krebs cycle in today's class itself. Okay, what is meant by Krebs cycle? What is exactly happening in the Krebs cycle? See here, so in just a glimpse of the revision of this yesterday's class. So in the glycolysis we have studied this can be happened in the aerobic respiration as well as the anaerobic respiration. Just take, we are taking now an aerobic respiration of glycolysis. Now in aerobic respiration of glycolysis, this glycose, which means this glucose, will be converting into 2-pyruvate. Okay, and this 2-pyruvate will be converting into 2-acetyl CoA, and this 2-pyruvate is the one converting into 2-acetyl CoA, and this particular reaction is called as a pyruvate decarboxylation. We are going to see about this equation. Now yesterday itself we have discussed about that equation, and after that 2-acetyl CoA is the one entering into the mitochondria and it is undergoing a Krebs cycle. Now what is exactly the Krebs cycle? The Krebs cycle will be the central metabolic pathway of aerobic respiration, where the acetyl group of acetyl CoA is completely oxidized to carbon dioxide and energy is captured in the form of NADH, FADH₂ and ATP or GDP. Here in a Krebs cycle the ATP will not produced, they producing GTP. See, the Krebs cycle, see, you should know that 2-acetyl CoA, this acetyl CoA will be the common pathway, common compound which has been, see here, carbohydrates, proteins, lipids. So in all these biomolecules, all the things have been converted at one point of time. All the carbohydrate molecule, protein molecule and the lipid molecule, that has to be converted into acetyl CoA. So this is the one, so this is the common intermediate of carbohydrate metabolism, protein metabolism and lipid metabolism. This can be expected in our exam. So which compound is acting as a common intermediate for all the metabolism? All the metabolism in the sense carbohydrate, protein and lipid. So the question may ask like you, whatever be the metabolism is, the common intermediate will be the acetyl CoA that has been taken place in the mitochondria in the means of Krebs cycle. So this is very important. Okay, so that's why we are saying this Krebs cycle is a central metabolic pathway of aerobic respiration. Definitely that has to be aerobic respiration. So in glycolysis we have studied that has been taken place in both aerobic respiration and anaerobic respiration, but in the Krebs cycle this is definitely will be taking place in the aerobic respiration. So here where the acetyl group of acetyl CoA is completely oxidized. Now this acetyl CoA is completely oxidized to carbon dioxide. Now here also a complete oxidation is taking place. That's why we are saying aerobic respiration. So oxidized to which compound? That is nothing but a carbohydrate, and energy is captured in the form of NADH, FADH₂ and ATP. And here in the ATP, in the Krebs cycle, ATP will not going to produce, that is a GTP will be produced. We are going to see about that. See, let me explain you what are all the basic information about this Krebs cycle. So this Krebs cycle is also called as a citric acid cycle. So the citric acid cycle is also called as a tricarboxylic acid cycle. Citric acid, it is also called as a tricarboxylic acid cycle, and it is also called as a Krebs cycle. Why it is also called as a Krebs cycle? Because it has been discovered by Hans Adolf Krebs. That's why the cycle named as the Krebs cycle. Now, where exactly the enzymes, where exactly the Krebs cycle is going to take place, that is mitochondrial matrix in case of eukaryote. So this is very, very, very important. Now, so this is what exactly the Krebs cycle. See here, the acetyl CoA has been formed from the 2 pyruvate. See, 2 glucose has been converted into 2 pyruvate, and this cycle is called as a glycolysis. And 2 pyruvate will be converting into 2 acetyl CoA, and this reaction is called as a pyruvate decarboxylation. We are going to discuss about that in the future. And in the acetyl CoA, see here, the 2 acetyl CoA, we are now discussing about 1 acetyl CoA. 1 acetyl CoA will be converting into what will converting into many of they, as this is a cycle. In the means of this cycle, they are producing many ATP, GTP and NADH and FADH₂. All those were the energy molecule. We are going to see about that. See here, acetyl CoA, this is the acetyl group, this is the acetyl group, and this is the CoA group. Here this CoA group has been eliminated. 6 carbon molecule formed from the acetyl CoA plus the oxaloacetic acid. See here, 1, 2, 3, 4. This is the 4 carbon molecule, which is nothing but an oxaloacetate. Oxaloacetate, this is the 4 carbon containing compound. This 4 carbon containing compound that has been mixed with, which means with 2 carbon containing compound, that is nothing but an acetyl CoA. Acetyl in the sense 2 carbon, here 4 carbon. So they, by eliminating the COA, that is a CoA, the CoA group has been eliminated. Now 4 carbon compound, that is oxaloacetate, and the 2 carbon plus acetyl group, they both combine together, they are forming a 6 carbon molecule, that is nothing but a citrate. Clear? Now the citrate has been converting into what? So as a result, now 1 carbon lost as a carbon dioxide here. After that, here 6 carbon. Now 1 carbon has been lost as carbon dioxide. They are going to give rise to isocitrate. We are going to first, as of now, I am explaining what are all the reactions they are taking place. In the next slide, that the citrate will be converted into which molecule, and further the name will be discussed in the upcoming slides. But just listen here, the citrate is a 6 carbon. As by eliminating 1 carbon, it has been that eliminating 1 carbon has been eliminated as carbon dioxide, and at this stage NAD plus is converting into NADH. So from the oxidized state, this NAD plus has been reduced as NADH. Now see here, 5 carbon. So 5 carbon molecule has been produced. Now in that 5 carbon, another carbon has been lost as the carbon dioxide. Now it results in the 4 carbon. Now again, so this NADH, here another NADH has been converted into NADH plus carbon dioxide. Now 5 carbon has been converted into a 4 carbon. Now in this 4 carbon, here the energy carrying molecule equivalent to ATP, that is nothing but GTP. Here the GDP plus PA, they are forming a GTP. Here NADH has been produced. Here also NADH has been produced, and here GTP has been produced. Now in the upcoming reaction, the FAD has been converted to the FADH, and this is also the electron carrier similar to the NADH. FADH₂. So here NADH, this will be the proper formula, and here it is FADH₂. Now malate, here also NAD plus has been converted to NADH. So how many NADH has been produced? 1, 2, 3. 3 NADH has been produced, 1 FADH₂ has been produced, and 1 GTP has been produced. So this is very, very important in an exam point of view. So this will be the complete Krebs cycle. Just see here, as oxaloacetate combined with acetyl CoA, acetyl CoA has been combined with this oxaloacetate, together they are forming a citrate. Here water has been included, here water has been eliminated. Now citrate has been converted to cis aconitase. In the next reaction the water is getting accepted, and now they are has been converted into isocitrate. Now from this isocitrate, here the NADH and NAD plus is converted to the NADH. One carbon has been eliminated as carbon dioxide. From isocitrate has been converted into alpha ketoglutarate. From this alpha ketoglutarate, it has been converted to the succinyl CoA. Whenever it is converting from the alpha ketoglutarate to the succinyl CoA, another NADH has been produced. This is very important. Now succinyl CoA has been converted to succinate with the help of the succinate CoA dehydrogenase. We are going to discuss about the enzyme in the upcoming slides. I didn't want to dump all the information in one slide itself, so that's why I have been separated the entire cycle. So the succinate has been converted to the fumarate. So whenever the succinate is being converted to the fumarate, FAD is being reduced to FADH₂. So this is also an electron carrier, again an energy molecule. Fumarate has been, with the addition of one water molecule, fumarate has been converted to a malate. Now this malate, whenever the malate has been again converted into oxaloacetate, one NADH molecule has been produced. So totally 3 NADH, 1 FADH₂ and 1 GTP will be produced in the citric acid cycle. Now what will be the starting molecule? Acetyl CoA. This acetyl CoA has been produced from 2 pyruvic acid. So 1 pyruvic acid, they produce 1 acetyl CoA plus carbon dioxide. Here 2 pyruvic acid converts into the 2 acetyl CoA plus 2 carbon dioxide. That's why the reaction decarboxylation, pyruvate decarboxylation. Now what are all the end products per cycle? One Krebs cycle, that is 1 acetyl CoA with 1 oxaloacetic acid, they are producing 2 carbon dioxide, 3 NADH, 1 FADH₂, 1 GTP. So this can be expected in our exam. Kindly make note of it. Very, very important. Now what are all the role of Krebs cycle? Why the Krebs cycle is very important in an exam point of view? See here, oxidizes acetyl CoA completely to carbon dioxide. So this is very, very important. What will be the reaction taking place in the Krebs cycle? As it is experiencing in the aerobic respiration, definitely oxidation will be taking place. Oxidizes the acetyl CoA completely to produce carbon dioxide. Now generates reduced coenzyme. So whenever the cycle is taking place, the coenzyme is also generating. What are all the coenzymes? NADH and FADH₂. Very, very important electron carrier, coenzymes and energy molecule. Whatever the thing we can say for these two molecules. Now it supplies electrons to the electron transport chain. What will be the important role of this Krebs cycle? That is the one. So this NADH and the FADH₂, both are the electron carriers. So they are carrying the electrons and they are donating to electron transport chain, which will be the final stage of the cellular respiration to supply electrons to the electron transport chain. So this NADH and FADH₂ plays a crucial role. Now they are producing a small amount of ATP directly. They are producing a small amount of ATP directly. This is also very important. Now this entry into the Krebs cycle, that is what the link reaction. So this is the link reaction. This is very, very important. Why? Because in the glycolysis, one glucose will be converting into 2 pyruvate. One glucose has been converted to 2 pyruvate, and 2 pyruvate, this pyruvate does not directly enter into the mitochondria, but instead it has been converted to the acetyl CoA. Only then it has been easily entered into the mitochondrial matrix. So before the Krebs cycle begins, the pyruvate has to convert into acetyl CoA. This is very important step. As it is converting the glycolysis process and the Krebs cycle, we are saying this particular reaction as a link reaction. 2 pyruvate will be converting into 2 acetyl CoA. Now for this conversion, which enzyme plays an important role, and the name of the enzyme will be the pyruvate dehydrogenase complex. And again, this pyruvate decarboxylation is also taking place in the mitochondrial matrix region. This is important reaction. This is a link reaction, and as a result of this pyruvate decarboxylation, they are producing 1 NADH, 1 carbon dioxide. See, just listen to me. See, per glucose, just listen to this reaction. This is very important step. 1 glucose will be converting into 2 pyruvate molecule, and this 2 pyruvate will be converting into 2 acetyl CoA plus 2 NADH plus 2 carbon dioxide. And what will be the name of the enzyme here? Pyruvate dehydrogenase complex. Very important reaction. And what will be the end product of this reaction can be expected in our exam. Now, so we are having totally 8 reactions in the Krebs cycle. How many reactions in the glycolysis? That will be 10. Half of the stages we are calling them as the preparatory phase, and the remaining phase will be the payoff phase. But in the Krebs cycle, it is involving in 8 reactions. 8 reactions together we are saying a Krebs cycle, and the first step will be the formation of a citrate. How the citrate is forming? Acetyl CoA, 2 carbon. See here, there will be no 2 acetyl CoA. 1 acetyl CoA, each acetyl CoA generally 2 carbon containing compound. This 2 carbon is mixed with this 4 carbon, that is the oxaloacetate. Together they are forming a 6 carbon, that is the citrate. So this is an important equation. This is the starting form of the starting step in the Krebs cycle. And which enzyme is mediating this reaction? That is citrate synthase. At this reaction, the acetyl CoA, the CoA molecule has been released in this particular reaction, and citrate has been produced. So this is very important with respect to this first step. And what will be the name of an enzyme? Citrate synthase. Now, the second step will be the isomerization. Isomerization, as I mentioned you in the previous class, see, glucose 6-phosphate and fructose 6-phosphate, both are the isomer. We can say that as an isomer. Why? Because structurally they are similar. 6 carbon, and here also 6 carbon, C6H12O6. C6H12O6, this is glucose, and C6H12O6, that is a fructose, and they are having a same molecular formula but different molecular formula. Same structural formula but different molecular formula. Why? Because in a glucose we can see the functional group aldehyde, and in the fructose we are seeing a functional group that is the CO, ketone group. That's what a difference between these glucose and fructose. Like in the same way, the citrate is also a 6 carbon, and isocitrate is also a 6 carbon. As it is an isomer, which enzyme is mediating this particular conversion? Aconitase. This is very important reaction. Now the third important step will be the isocitrate is getting converting into alpha ketoglutarate. Alpha ketoglutarate. And see here, the 6 carbon, here the 6 carbon, that is citrate, is converting into isocitrate, and from the isocitrate it has been converting into 5 carbon, that is alpha ketoglutarate. And which enzyme is mediating here? Isocitrate dehydrogenase. As I have explained you before, wherever we are seeing the enzyme which is end with the name dehydrogenase, there definitely NADH will be produced. NADH, or it will be FADH. Definitely these molecules will be produced whenever the enzyme dehydrogenase is involving in any particular reaction. And as it is an energy molecule, As it is an energy molecule, oxygen is getting utilized here. We are saying this is a first oxidative decarboxylation. See here, oxidative decarboxylation. Why we are saying decarboxylation? Carbon dioxide has been released here. Carbon dioxide and NADH has been produced. That's why the name oxidative decarboxylation. So in the question, if they asked first oxidative decarboxylation in the Krebs cycle will be taking place in which of the following steps? We know that will be isocitrate whenever it is converting into the alpha ketoglutarate with the help of an enzyme isocitrate dehydrogenase. This is the third step. And the fourth step, the second oxidative decarboxylation. This will be the second oxidative decarboxylation reaction, that is alpha ketoglutarate, that has been converting into succinyl CoA, which means the 5 carbon will be converting into a 4 carbon. And what will be the name of an enzyme? Alpha ketoglutarate dehydrogenase complex. Again there will be the involvement of dehydrogenase enzyme. So definitely there will be the production of NADH. And here 5 carbon has been converted to 4 carbon, which means 1 carbon has been removed, that has been produced as carbon dioxide. So the second oxidative decarboxylation will be taking place in the fourth step. So kindly make note of it. This is also another important step. Now this fifth step will be the substrate level phosphorylation. Substrate level phosphorylation, the succinyl CoA will be converting into succinate. So here also the CoA has been removed. So the CoA has been removed with the help of the succinyl CoA synthetase. So whenever the synthetase enzyme has been hexokinase, whenever the enzyme has been end with the suffix word kinase, definitely they are involving in adding the phosphate group. Like in the same way dehydrogenase, definitely there will be the production of this NADH, no doubt about that. So in that line, if any enzyme will be end with the word synthetase, again there will be the production of ATP or GTP. One ATP molecule or GTP molecule or UTP, CTP, whatever it is, whenever there will be the enzyme which ends up with the word synthetase, definitely going to produce this ATP. And here in Krebs cycle it will be GTP. So here a substrate level phosphorylation will be taking place in the Krebs cycle. Okay. So this is very important step. And the sixth step, that is oxidation. Here the succinate will be converting into fumarate. Here the succinate has been converting into a fumarate with the help of the succinate dehydrogenase. So again the dehydrogenase enzyme has been involved. Either NADH or FADH₂ will be produced. Here FADH₂ has been produced. So fumarate has been produced and the FADH₂ is also produced here. And the seventh important reaction, the name of the reaction will be the hydration reaction. Here the fumarate will be converting into malate, and with the help of an enzyme called as a fumarase enzyme. And here oxidation reaction. Step 8, this will be the last important step of the Krebs cycle, that is oxidation reaction. Malate has been converting into oxaloacetate with the help of the malate dehydrogenase enzyme. So whenever there will be the involvement of dehydrogenase enzyme, definitely there will be the production of this NADH. And oxaloacetate is regenerated, allowing the cycle to continue. Again the oxaloacetate has been together with the acetyl CoA, it has been converted into a citrate, and citrate to isocitrate, and isocitrate to alpha ketoglutarate, and alpha ketoglutarate to succinyl CoA to the fumarate, and fumarate to malate, and malate to the oxaloacetate. Now this can be expected in the exam. This is very important chart. Net energy yield per 1 acetyl CoA. See here what we have discussed, 2 glucose will be converting into 2 pyruvate, isn't it? And 2 pyruvate will be converting into 2 acetyl CoA. right? Now one acetyl CoA is continuing the Krebs cycle, another acetyl CoA is also continuing the Krebs cycle, but you should know, see, this is not 2 glucose, I'm extremely sorry, this is only 1 glucose. 1 glucose will be converting into 2 pyruvate. 2 pyruvate will be converting into 2 acetyl CoA, and each acetyl CoA is undergoing a Krebs cycle. So, but now we are calculating only for 1 acetyl CoA, and after that we are multiplying into 2, because 2 acetyl CoA is involving in the Krebs cycle, and that 2 acetyl CoA has been produced from only 1 glucose. Don't confuse in this statement. Let me explain you again. See, glucose will be converting into 2 pyruvate, and this cycle is called as a glycolysis. And 2 pyruvate has been converting into 2 acetyl CoA. And acetyl CoA, this will be entering into the Krebs cycle. See here, one acetyl CoA is continuing the Krebs cycle, another acetyl CoA is also continuing another cycle, but both acetyl CoA has been produced from how many glucose? Only 1 glucose. This can be expected in our exam. So one acetyl CoA, if they are entering into the Krebs cycle, they are going to produce 2 carbon dioxide and 3 NADH. And just make sure you know all the area where exactly this NADH has been produced, and FADH₂, one will be produced, and 1 GTP molecule will be produced. So this is what with respect to 1 acetyl CoA. What if 2? See here, if we can calculate into 2, which means in 2 into 2 it will be 4. Because here for 1 acetyl CoA, another acetyl CoA will be here, and here also 3, it will be 6, and here again 1, it will be 2, and here also 1 GTP 1. So totally how many number of carbon dioxide, NADH, FADH₂, GTP will be produced in the Krebs cycle for 1 glucose? Then you have to add all these things. That is 4, 6, 2, 1. You have to understand this calculation. Yeah. So per 1 glucose, 2 turns of a cycle, then what will be the result? 4 carbon dioxide, 6 NADH, 2 FADH₂, and 2 not ATP, GTP will be produced. Yeah. So like I said in the yesterday's class, 1 NADH₂, 1 NADH will be approximately equal to 3 ATP, but now they are saying 2.5, and as a round value we are saying 3, and 1 FADH₂, we have said approximately it will be equal to 2 ATP, and here the exact value will be 1.5. Just remember this as a 3 and this as a 2. Don't confuse. Now listen to me very carefully. So per acetyl CoA, 1 acetyl CoA, they are producing 3 NADH, 1 FADH₂, 1 ATP. And whenever we are calculating this NADH and FADH₂ into ATP, we have to multiply this 3 into 2.5. Then we can say that into 7.5. 1 FADH₂ is approximately equal to into 1.5. So 1.5 ATP, and 1 GTP molecule has been produced. We can calculate that into 1. So totally 7.5 plus 1.5 plus 1 together we can say that as a 10 ATP per acetyl CoA. Per acetyl CoA totally 10 ATP. And if we calculate this for 2 acetyl CoA, then 10 into 2 is equal to 20. So this is how we have to calculate. So how many ATP has been produced from the Krebs cycle? That will be 20 ATP. So this is what the complete information about a Krebs cycle, and what will be the overall significance of this Krebs cycle. Central metabolic hub. This is the common, they are producing a common intermediate, that is nothing but an acetyl CoA. So this is common intermediate for carbohydrate metabolism, fat metabolism, and protein metabolism. For all these metabolism, acetyl CoA, at the end they have to convert into acetyl CoA, and the Krebs cycle will be continuous. Only if the Krebs cycle has to continue, definitely there has to be the production of acetyl CoA. Provides the precursors for amino acid, heme, nucleotides, and gluconeogenesis. So these are all the precursors, very important. Now, so in the glycolysis process, we have studied about the important enzyme, which is a regulatory enzyme. We have studied about the most important key enzyme, that is a rate limiting enzyme. So that is phosphofructokinase, PFK. So here also we are having three key most important regulatory steps and three major control points in the Krebs cycle. Now what are all the enzymes and what are all the regulation? Citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. These are all the three most important enzymes which is regulating the entire Krebs cycle. So the citrate synthase has been inhibited by ATP, NADH, citrate. So these are all the three important inhibitors which are inhibiting the citrate synthase. Very, very important. So what do we have discussed for phosphofructokinase? They have been inhibited by AMP and ADP, which means activator. These are all the activators. With the presence of ATP, this phosphofructokinase is going to inhibit. So that's the thing. Like in the same way, the citrate synthase is inhibited by ATP, NADH and citrate. Isocitrate dehydrogenase is activated by ADP and calcium, and alpha ketoglutarate dehydrogenase has been inhibited by NADH and Succinate CoA. So these are all the three most important control points, control enzymes and their regulation. So just remember all these things. No other way you have to memorize this. Now, what will be the why we are saying this particular amphibolic nature of the TCA cycle? See, I have explained you the. See here, I have explained you about the metabolism. Metabolism. So in metabolism we are having two important ism, that is catabolism and anabolism. Catabolism and anabolism together we are saying that is a metabolism. Anyhow they are going to break something or they have to make up something. So here a complex molecule has been broken down into a simpler molecule, that is called as a catabolism. Next, anabolism, as they are breaking the smaller molecules has been together, they are making up any complex molecules, and that is nothing but an anabolism. And in the Krebs cycle, both catabolism and anabolism is taking place. That's why we are saying that as an amphibolic nature. Very, very important. This can also be expected in our exam. So what are all the things we are discussed in our class? Definitely all those points are in an exam point of view. None of the information is apart from this exam track. We are going in the correct track of an exam, so kindly go through all the videos. See here, what are all the intermediates? Intermediate means what? Citrate, alpha ketoglutarate, Succinate CoA and Oxaloacetate. See, each intermediate, they are involving in biosynthesis or some other process. So what are all those? Citrate. This is very important for the synthesis of fatty acids and sterols. Alpha ketoglutarate, they are involving in synthesis of glutamate and other amino acid. Succinate CoA is very important for the synthesis of heme, and oxaloacetate is important for the synthesis of aspartate and glucose. They are not only producing ATP, and some of the intermediates is also necessary for the synthesis of some useful products. So this can very, very important examples. Now, see, anaplerotic enzyme reactions. What is mean by anaplerotic reactions? Reaction that replenishes TCA intermediate. So here, so not only the Krebs cycle is following, the citrate is converting into isocitrate is converting into something. So this reaction is not only just forming of a product and the reactant, each intermediate is also involving in some other synthesis. That's why that's what an anaplerotic reaction. So the most important reaction will be the pyruvate plus carbon dioxide. They are forming an oxaloacetate with the help of the pyruvate carboxylase enzyme. So this is very important. Yeah. What are all the key important enzymes? So just now we have studied. So here whenever we are discussing about this electron transport chain, we are discussing about 4 important complex like complex 1, complex 2, 3 and 4, and the complex 2 of electron transport chain is nothing but this succinate dehydrogenase complex. Very important statement, just remember that. Now see here, these are all an important point of view with respect to an enzyme. The same thing, these are all an important point, kindly go through it, the same thing what we are discussing in previous slides. And this is the comparison between this glycolysis and Krebs cycle. This is taking place in the cytosol, and here it will be in the mitochondrial matrix. And glycolysis, the oxygen not required directly, and it is indirectly required for that's why we are saying that as an aerobic respiration. And here it is taking place in both aerobic as well as anaerobic respiration. And here the main function will be the conversion of glucose into pyruvate, and here there will be the acetyl CoA has to be completely oxidized to the carbon dioxide. Now ATP has been produced in glycolysis will be 2 ATP, and in the Krebs cycle 2 GTP. For 2 acetyl CoA, they are specifically saying about ATP, but we are having NADH2 and FADH2 also, but here specifically asking about this ATP. Yeah, the same thing, I don't want to repeat the thing again. So let's move on to a question. The Krebs cycle occurs in which part of the cell? Krebs cycle will occurs in which part of the cell? That is mitochondrial matrix, no doubt about that. Because the enzyme, see here, very important explanation, all enzymes of the Krebs cycle are located in the mitochondrial matrix except a succinate dehydrogenase, because the succinate dehydrogenase complex is important in Krebs cycle, but that is present in the inner mitochondrial membrane. So this can also be expected, kindly make a note of it. And which enzyme catalyzes the first step of the Krebs cycle? First step, that is citrate synthase from the acetyl CoA plus oxaloacetate. Together they are forming a citrate with the help of the citrate synthase enzyme. Now, how many molecules of NADH are produced per turn of Krebs cycle? Per turn, how many NADH? Just pause the video and answer. Let me read this question again. How many molecules of NADH are produced per turn of a Krebs cycle? So per turn, how many molecules of NADH will be produced? 3 NADH. They are not asking about 2 turns, because 2 acetyl CoA will be produced. For 1 turn they are producing 3 NADH. For 2 acetyl CoA, that is 2 turns, they are producing 6. Now, which intermediate of the Krebs cycle is directly involving in heme synthesis? Heme synthesis, definitely there will be the involvement of the succinyl CoA. And which enzyme of the Krebs cycle is also a component of the electron transport chain? So in electron transport chain, we are having a 4 complex, and the complex 2 is nothing but the succinate dehydrogenase complex. And the conversion of succinyl CoA to succinate produces succinyl CoA to the succinate. So you have to know what will be the name of an enzyme. Succinyl CoA dehydrogenase complex. So definitely there will be the production of GTP. Once again, let me explain you with the help of this picture. Yeah, see here, succinyl CoA is converting into a succinate, and what will be the name of an enzyme? Succinyl CoA synthetase. If the synthetase enzyme is involved, definitely there will be the production of this GTP. So that's what asked in the question. Yeah. How many carbon molecules has been released per acetyl CoA in the Krebs cycle? It is not asking 2 acetyl CoA per acetyl CoA. That will be definitely 2. to carbon dioxide. Which of the following act as an allosteric activator of the isocitrate dehydrogenase? I have explained you what is allosteric enzyme. So allosteric enzyme, in the sense there is a substrate binding area, that is an active site. Together they are also having an additional site where the activator is going to bind. Here the activator will bind or inhibitor will bind. So here they are asking about the allosteric activator. So that's what an isocitrate dehydrogenase. I have explained you this allosteric enzyme in the enzyme class itself, and that will be the ATP and ADP. And the Krebs cycle is described as the amphibolic because why it is said as an amphibolic? Why? Because they are both involving in both catabolic and in the anabolic pathway. Now, how many ATP equivalents are generated per ATP per acetyl CoA during the Krebs cycle? So, how many ATP equivalents? Here they are not asking how many ATP molecules. ATP equivalents per acetyl CoA including NADH and FADH₂. So, totally 10 ATP will be produced. So, this calculation we have discussed in, before itself, previous slide itself. So, that's all about the Krebs cycle. Kindly go through it. We'll meet you in the next class, and we'll finish up the remaining cellular respiration. Thank you so much.